Well, welcome everybody to the Midland and West of England Great Dane Club first Zoom seminar. I've great pleasure in introducing Neil O'Sullivan, an expert in DNA sequencing, statistical analysis, I've got to put my teeth in, genomics, genetic analysis, genotyping, population, genetics, genetic diversity, and next generation sequencing, as well as being a very sweet and kind individual giving us his time today. Neil was born in 1963 in Cork, Ireland, so he's 10 years younger than me, into an active dog family and enjoyed showing from a young age. The first dog he owned with his sister Margaret was a brindle Dane, which unfortunately developed wobblers at 12 months, only surviving 2 more months, yet inspiring the 14-year-old Neil's interest and future in genetics. Neil completed a PhD in genetics by 1991, but had already published a peer-reviewed paper on Harlequin genetics in 1989 with the late Roy Robinson. His passion for helping Great Dane breeders using his research is ongoing. As a breeder, Neil has enjoyed success across the colours and, along with his husband Gerard, also breeds soft-coated Wheaten Terriers, Briards and show Kerry Blue Terriers. In his professional life, Neil is a consultant in poultry genetics with an emphasis in producing pullets in cage-free environments and cage-free production. Neil chairs the committee developing the updated Great Dane Club of America colour code and is particularly interested in the application of DNA and genomic advances in Great Dane breeding. Our secretary, has sent you all a PowerPoint presentation which will hopefully be up on our website as well next week. If you have any questions after this seminar or during the seminar, please email them to our secretary, secretary@midlandandwestgdc.org, so that Neil can present them to Dr. Neil at the end. Over to you, Neil.
Thank you. And it's a great pleasure to give another seminar for the Midland and West Great Dane Club. I believe I gave one in the 1980s, so a long time ago. I entitled the talk, The Merle Gene, the gene we didn't know we needed. When I was a young man in Great Danes, which is a little while ago now, it was normal practice to euthanize Merles and Whites at birth. And so they really were very rarely seen. And it was considered, there was considerable pressure almost to hide your Merles and hide your Whites. So I became very interested in this particular gene and why it was being shunned. So you'll see as we go through the presentation how that all came about. So on to the next slide, and it's only one of 3 slides you're going to hate, but it's important that you understand the technical background. This particular gene that we call Merle, it resides on chromosome 17. And what it is a change within this pre-melanosome protein. So a protein that is designed to help the melanin be built. So by having a defect in this gene, and the defect is called a short interspersed nuclear element, but associated with that, there's a repeat sequence, which we call a poly T-tail, okay? And I've got a picture of it for you later. Non-Merle is the normal form, and it's actually a homozygous recessive. So once this insert goes in, it disrupts this protein, okay? And that's why it's dominant, because the disruption of the process of making melanin occurs. Almost all mantles and blacks are normal homozygous non-Merle. Merles and Harlequins are both heterozygous for the gene, meaning that they carry one copy of the dominant Merle gene and one copy of the non-Merle gene. This insert, the sign, its variation in length has been studied relative to the expression of Merle, because you're all familiar with the fact that there seems to be, a Merle is not the same. There's quite a bit of difference in the expression of Merle, and we'll talk about that some more. On the next slide, I actually have the picture. So my husband said to me, Neil, what's an extron? So an extron is a piece of DNA that flanks an active gene, but it is non-encoding. It's like a spacer. So it's not junk DNA. It has a function, but its function is to space the genes out on the genome. So what happens is, within this pre-melaninome protein gene, you have this sign, this element in there. And I actually have it upside down because that's the way it gets in there, okay? And you'll see there's a section called poly T. This is where the TTT is repeated. And the variation in length of this poly T is what creates variation in expression of the Merle gene. All right, on to the next slide, and we'll talk about some of this variation. One last point: if a dog carries two copies of the Merle gene, in Great Danes or whites carry two copies of the Merle gene, but so do our Merlequins. Okay. And we'll talk a little bit later about what the difference there is. So because of the difference in length of the sequence, and the bigger the length, the more disruption there is of the production of pigment. So some of you have probably run across a mantle or a black that actually does carry for Merle. This is typically what we call a cryptic Merle. It has a very short poly T tail. Some of you might have actually seen, my sister has a very flashy marked Harlequin bitch, mantle bitch. But when we bred her to a Harlequin dog, you would expect no white puppies, and there was a white puppy in there. And that's how we found out she actually was cryptic Merle. So these cryptic Merles are very hard to tell from a regular mantle. There's a form we call atypical, and that's a dog typically who looks like a mantle or a black, but you can find some Merle on it, a small amount of Merle on it. And that's what we call the atypical. And its poly T tail is a little bit longer than the cryptic. The next one in the sequence is the most common, the normal Merle gene that gives us the typical Merle pattern we recognize, or phenotype. And then there's one unfortunately named Harlequin mantle. The term, it's not a true mantle, but what it has is it's got a very long T-tail, and it manages to wash out all of the background normal gray color on a Merle. So at the bottom of the page, you'll see the terminology. M is for normal Merle. M with a small superscript C is a cryptic Merle. MA superscripted is the atypical Merle. And MH superscripted is the so-called Harlequin Merle. and then the small letter M is used to designate a non-Merle. That's not a no Merle, by the way. That was my typo. Okay, so where the mystery began from was mating harlequin to harlequin produces many different colors. And so for me as a young man, that question puzzled me greatly. Why couldn't we breed harlequins true if we kept mating harlequin to harlequin to harlequin generation after generation? So that's what I went about studying. And if we look at what actually is occurring today from the knowledge we have, so I've genotyped out for you here what a harlequin is. And in this case, just not to complicate the story, my harlequin parents in this example on this slide, they don't carry any undesirable colors. You know, they're not going to carry for fawn, or they're not going to carry for black and tan or chocolate. So how we designate that is at the A locus or the agouti locus, you'll see that typically a Great Dane is AY. The only variant we see in Great Danes at that loci is black and tan, or tan points strictly, and that would be an AT. They do occur, they're very rare. In Great Danes, we very rarely see chocolate, so they're normally homozygous dominant black at the B locus, and we rarely see chocolate, which would be a recessive. This harlequin, just to save complications, is a black harlequin, so it does not carry any dilution gene. At the E locus, many harlequins are fixed for non-masking. We can tell as harlequins and mantles get older if they carry a masking gene, because you'll see more intense graying in the face, whereas many harlequins don't tend to gray very much in the face at all, and those ones happen to be homozygous for the small e. All harlequins carry one copy of the harlequin gene and one copy of the non-harlequin gene. At the K locus, this example, we've got dominant black, and that would be the common thing there. It would be at that K locus, if they carried fawn, we'd have a KY superscript. At the Merle locus, you can see we got dominant Merle and non-Merle. And then just to complete the picture, since most harlequins tend to be fixed for what's known as Irish spotting, but we call that mantle in our breed, that's at the spotting gene, that's the SI, and the I superscripted there stands for Irish spotting. So the only thing our two harlequin parents are segregating here for are at the H loci and at the Merle loci. So each parent is going to produce identical gametes, so that would be the sperm and the eggs. And a sperm or an egg only carries one copy of each of the chromosomes. So I've listed that out as one gamete they could produce is with dominant Harl and dominant Merle. Another is the dominant Harl with the recessive Merle. Then the recessive non-Harl with dominant Merle. And then the two recessives, non-Harl, non-Merle. And I put a note on here, you know, matings of Harl to Harl, we do need to proceed with caution, because this mating does produce white and merlequin progeny. And as we'll get into a little bit later, there's some health issues associated with that. So on the next slide, I've done a Punnett square, and this is of the harlequin to harlequin breeding. So one parent is contributing these different gametes on the very top line, and on the column, first column, we see the other parent's gamete contributions. And you can see, although there's a total of 16 possible combinations, only 12 of these are viable because 2 copies of the dominant harlequin gene causes an in utero lethal. So 4 of the possible combinations never actually implant into the uterus, and then the other growing fetus do implant. So we can see the combination produces 2 whites and 1 merlequin, and I put that in there in red to designate that those animals represent a risk for having a lack of pigmentation in the inner ear, or a lack of pigmentation on the retina, and so leading to microphthalmia and blindness. So that's why they're marked in red. The other dogs who are perfectly healthy, the 4 harlequins we see here, the 3 mantles and the 2 merles, those all are perfectly healthy animals. So on the next slide, I just put a summary of what harlequin to harlequin typically produces. So the average result, this is not what's going to happen in an individual litter. Why? Because statistically, each combination is a sample. There's a random admixture. So in some litters, you may get more than the 33% harlequin. But if you average out over many litters, as we did, the average results are 33% harlequin, 25% mantle, 17% merle, 17% white, and 8% merlequin. So on to the next slide. So we can now say definitively that harlequins are all merles. Also, most harlequins typically have a black mantle pattern overlay on them as well, but they definitely carry 1 copy of the merle gene and 1 copy of the harlequin gene. As I said, 2 copies of harlequin in utero is lethal. Whites are homozygous merles, but you can see they carry 1 copy of the harlequin gene. So the harlequin gene expresses more whitening, and basically you don't see any of the gray at all. Whereas the merlequin, who doesn't carry it, you see these large, they're typically torn patches of merle on the dogs, but they're definitely a merle and white patterned dog. Unfortunately, though, if their white areas fall in the inner ear, then there's no pigment placed down in the inner ear, and those dogs are deaf. Or if there's no pigment on the retina at the back of the eye doesn't grow to its normal size, and because it's apigmented, the dogs are blind. So that's why when I said we need to proceed with caution with these breedings, there's a definite health risk associated with being white or being merlequin. All harlequins are therefore designated as these dual obligate heterozygotes, meaning they have to be heterozygous and they have to be so at 2 different genes, which is why they're very complicated to breed. They will never breed pure because you've got this requirement for heterozygosity of 2 different genes. I think if we think about the bigger picture, though, both chromosome 9, where the harlequin gene is, and chromosome 17, where the merle gene is located at, those 2 chromosomes are heterozygous in every harlequin. And I think that may contribute to the diversity that we see imposed upon harlequin all of the time. Anybody who's spent any time knows that those dogs, they have a particular vitality to them. And I think it's this requirement that always injects quite a bit of variation for them. I did put a note at the bottom. Many countries have banned the breeding where both parents carry Merle. The approach that the Great Dane Club of America is trying to take is we would like to discourage people matings where both parents carried Merle, but Americans being Americans, it's very hard to impose a rule on them. They will buck. But I believe most of the FCI countries in Western Europe do not permit Harlequin to Harlequin or Harlequin to Merle breeding. Okay, on to the next slide. Very common mating we see is mating a Harlequin to a mantle. Both parents, again, I don't want to make things complicated here. Both parents don't carry any. And in this case, the mantle does not carry for Harlequin. So the Harlequin genotype we went through earlier. The mantle, I'll just highlight where the difference is. The mantle is homozygous for the non-Harlequin gene, and it's also homozygous. My trusty glass of water. Okay, so in this case, the Harlequin is segregating for both Harlequin and Merle, but the mantle is not. So the gametes are the 4 gametes we saw earlier that can be produced by the Harlequin parent. And the only gamete this mantle can produce is one with non-Harlequin, non-Merle. And on the next slide, I just have the Punnett square of how that works out. So you can see on the top line, I have the 4 gametes coming from the Harlequin, and on the first column, the 1 gamete coming from the mantle. And as we combine them, we can produce 1 Harlequin, we can produce 2 mantles, and 1 Merle. Obviously, a very clean mating in terms of no complications here with colors that aren't recognized, except, of course, in the UK at the moment, the Merle has not been recognized. But we've seen it proceed quite quickly through the continent and through North America, the approval of the Merle. So the Harlequin to mantle, where this mantle does not carry the Harlequin gene, gives us 25% Harlequin, 50% mantle, and 25% Merle. Again, remember an individual litter is a sampling of how it can occur, so it can deviate very significantly from the average across many matings. Here's the second on the next slide, the second Harlequin mating. This time the mantle does carry for Harlequin. So the only difference we really see here is that now the mantle has 2 different gametes, one of which does carry for Harlequin and one of which does not. So as we proceed on to the next slide, you'll see that Harlequin to mantle breeding, where we're now carrying for Harlequin, because both parents can contribute a Harlequin allele, we see that out of the possible 8 combinations, 2 are in utero lethal because they are homozygous for Harlequin. Now you'll see we have, of the 6 possible phenotypes, 2 are Harlequin, 3 are mantle, and 1 is Merle. So on the next slide, you'll see that our percentage Harlequin is now up to the same as we were getting from a Harlequin to Harlequin breeding, but without the danger of white, without the danger of Merlequin. and we have 50% mantle and 17% Merle. So you're starting to see a pattern here where if we do these combinations correctly, we can very reliably produce 3 colors that are healthy and can be accepted in the show ring, which would be Harlequin, Mantle, and Merle. And we can look at strategies where we avoid producing white and Merlequin. The next mating, I want to show you what happens if you mate a Harlequin to a Merle. Again, I've left the Harlequin. I haven't complicated things with any undesirable color yet. So our Harlequin is the same genotype we've looked at. This time, our Merle is, of course, homozygous for non-Harlequin, but is heterozygous at the Merle loci. So again, we have the 4 gametes we're familiar with coming from the Harlequin, and this time it's at the Merle gene that the Merle is segregating. However, this is again a breeding that has the potential to produce white and Merlequin, so we should proceed with caution. On the next slide, you'll see the result of mating Harlequin to Merle. We have produced a white offspring, and we have produced a Merlequin offspring. We also have 2 Harlequins, 2 Merles, and 2 Mantles. So the summary on the next slide of that is we have 25% Harlequin, 25% Mantle, 25% Merle, 12.5% white, and 12.5% Merlequin. So how can we breed our Merles? So the next slide shows us a Mantle to Merle breeding. In this case, I have a Mantle which does carry for Harlequin. So our Mantle is heterozygous at Harlequin, and our Merle is heterozygous at Merle. So they each can produce 2 different gametes. And on the next slide, we'll show how those combined. We got 1 Harlequin, we got 2 Mantles, and 1 Merle. So if we look at a summary slide of that, it's 25% Harlequin, 50% Mantle, 25% Merle. So you're beginning to see now why people are testing their mantles for whether they carry for Harlequin. I'm also recommending that if the lab you're testing at offers Merle testing, to do so, because you might have a cryptic Merle in there. Okay, the next mating is a Mantle to Merle. Neither parent carries any undesirable genes, but in this case, the Mantle does not carry for Harlequin. So the only gene segregating is the Merle gene. If we go on to the next slide, you'll see that this Mantle to Merle breeding produces Merle or Mantle only. And on the summary slide, that's approximately going to be 50% Mantle, 50% Merle. Some of you have probably seen Mantle to white breedings done. And I have a few examples of that. On the next slide is a white and Mantle being bred together. In this case, both the Mantle and the white carry for Harlequin. And of course, the white is homozygous at the Merle. And the Mantle is, of course, homozygous for non-Merle, but heterozygous for Harlequin. So on the next slide, we'll see the results of that. Because both parents carried a Harlequin gene, we do have 1 in utero lethal potential combination, but the 3 viable combinations, 2 of them result in Harle and 1 result in Merle. So in this particular Mantle to white breeding, the summary chart would show us that we have 66 percent Harlequin and 33% Merle. So if you want to produce a lot of Harlequins, this particular combination maximizes that. Remember, in a Harlequin to Harlequin breeding, we only got 33% Harlequin, and we got lots of messy things like whites and Merlequins. So what about a mantle to Merlequin breeding? Remember, the Merlequin differs from the white only in that it does not carry for Harlequin. In this particular case, we actually don't have any segregating genes occurring. But the only difference is the combination of the parents. So if we flip to the slide with the Punnett square, you'll see that the mantle was contributing a non-mantle gene, and the Merlequin was contributing a Merle gene. So we get a litter that's 100% Merle. And that's shown on the next summary slide. Okay, let's talk about doing a Merle to Merle breeding. So again, both parents not carrying any undesirable color genes, and our Merles are heterozygous for Merle. So what's segregating here in both parents is Merle. Again, I say proceed with caution because this particular combination can produce white progeny. And on our slide with the Punnett square, we see both parents contributing a dominant Merle gene, giving us one white. And in the combination, we have two Merles and one mantle. So in the summary of that, we have 50% Merle, 25% mantle, and 25% white. And that's a fairly high percentage of whites to have in a litter. Let's talk about mating a Merle to a black. So both parents, again, not carrying any undesirable color genes. But this time where the segregation is occurring is obviously for Merle, but also in the spotting gene. You can see the black is homozygous for solid color. So in other words, this black would have very little white on it at all, maybe on the toes or a spot on the chest, but nothing more than that. So now we see a little bit of a different combination with the gametes here, because our black is contributing a solid, and our mantle, of course, is contributing Irish spotting. So the contributions coming on top from the mantle, and on the first column coming from the black. So now you can see we'd end up with a litter with Merles and blacks. But in this case, because they both are solid, we don't see any mantles occurring in this breeding, even though the Merle was contributing mantle. And that's how we get a solid Merle, is when we have one copy of the non-spotting or dominant solid color. So a summary of that is 50% black and 50% Merle. And as I said, the black may be with or without white on the toes and chest, and the Merle will be solid, again, with or without white on the toes and chest. And on the next slide, I think is the story of how I got interested in all of this. Many of you probably remember Anne Bodie when she worked for Margaret Everton in the 1980s. And Anne had a blue bitch, which to everybody's surprise carried a Harlequin gene. Margaret had been dabbling in the 80s when Sven Peterson was in the UK with some Harlequin breeding as well. And Sven had used some of her blues in his breeding program. And so Margaret had done a couple of Harlequin litters. And I happened to be there staying with Margaret and Anne when this happened. Anne was out kennel cleaning, and the blue bitch got out and had a tryst. And Anne came screaming into the house, asking to assist with an already tied pair. It wasn't going to happen. So anyway, in the resulting litter, there was a Harlequin puppy. And to say that, people were shocked by that is an understatement, because at that time, people thought you had to have a Harlequin parent in order to produce a Harlequin puppy. So what I started to do was collect the records from the breeders in the UK in the 80s. And I must say I had tremendous cooperation and a couple of breeders in Ireland as well. And that allowed me to build up the database that I was able to analyze so that I could produce my first research paper, which was on the inheritance of Harlequin in the Great Dane. So let's look at this Merle to blue breeding. And this is actually, what I would summarize as the genotypes of the two individuals involved. The Merle dog is just segregating at the Merle gene. And that Merle dog had a mantle pattern as well. He was very, very flashily marked dog, very handsome dog. Blue bitch segregated for Harlequin, as by the way do many of our fawns and brindles. It's not uncommon at all for dogs that have nothing to do with Harlequin breeding in the last 30 to 50 years to still carry a Harlequin gene. But the blue, of course, was not segregating for Merle. So it's a little bit of a complicated mating, because we've got segregation occurring at the dilute for the blue, at the Harlequin, at the Merle, and at the spotting gene. So we've got a couple of different gametes that both the Merle and the blue were contributing. So on to the Punnett square. And you can see the result of that is indeed 1/4 of the litter carried for Harlequin. You'll notice I've used an orange color here, and that orange is just telling you to proceed with caution, meaning in breedings like this, you should test the progeny and also test potential mates. Well, in the UK, producing a blue Harlequin is fine for your standard. In most parts of the world, outside of the UK, I think Australia and New Zealand, blue Harlequin would be disqualified. Harlequins are designated only as black-white with some Merle on them. So in this breeding, indeed, this is how we produced a Harlequin, from a blue and a Merle. We also, the letter had blacks and Merles in it as well. So the average result for this particular breeding was 50% black, and most of those blacks had almost no white on them at all. Same thing with the Merles, they were solid Merles. And there was one Harlequin male in the litter, and he did have some color on the neck and on the front. Don't remember if he had it on his legs or not. But Harlequin dogs that don't have that mantle pattern superimposed on them, you'll know that they carry for solid if you see significant amounts of markings on the neck, on the front, and on the lower legs. So, next slide. Now I can definitively make the statement Harlequins cannot be bred without the Merle. And clearly we have no scientific reason to discriminate against Merle in her breed, because we can manage her breedings so that the presence of the Merle gene, we avoid producing homozygous Merles, which are whites, and Merlequins, which are the only danger to her breed. And you know, if you talk to a Daxi breeder, for example, who produces dapple, they would never breed two dapples together. You talk to your collie friends, you would never breed two blue Merles together. So it's a rather sensible approach that breeds with the Merle gene have taken. In our case, because I think we had Merle as sort of our dirty secret behind our Harlequin breeding programs, I think we developed a rather bad attitude towards it. And as I said, as a child, I remember, you know, actually people putting white puppies down at birth and Merle puppies down at birth. Okay, next slide. So my statement is, and it's a strong one if you think about this implication for the breed in the UK, without Merle, we have no Harlequin. And so this is a very strong justification, and the justification that was used in the US as we went about approving the Merle was we could not have our Harlequins without having our Merles. And there was no reason to then deny perfectly good breeding stock from being able to go in the ring and be evaluated in our normal peer review evaluation system that works so well in dog breeding. So Harlequins are actually modified Merles. The Merle gene it only acts on the dilute gray areas, and it actually doesn't do a perfect job because on every Harlequin, you'll find anywhere from a small area of Merle left to, you know, we had a beautiful bitch in America called Paige who had it on the show side of her head, but she was exquisite and won the nationals. So imagine the Harle gene as being the one that affects almost only the gray pigmentation that we call Merle. So a couple of other things to think about is in the Irish spotting. We're working in the US to have a test for the Irish spotting gene, the mantle gene. We actually know what its sequence is. We just have to have the test work effectively. So we'll be able then to distinguish a solid from an Irish spotting from the piebald, which is the other one we have in our gene pool. Piebald is foreign to our gene pool. If you don't know what I mean by piebald, think of a typical pointer color. So these are circular patches of color, be that black or brown or liver on a pointer, but they're very rounded, solid color. They're not the jagged, torn patches that we have due to our Harlequin and Merle genes. A high percentage of mantles and Harlequins that you test will actually carry piebald. So that is about it, and hopefully you've accumulated lots of questions for me.
Neil,
and if you'd like to go back to explain anything in more detail, it'll be my pleasure to do so.
Okay, Neil, I've had one email me from Tammy, which I've forwarded to you because I think you probably need to see the diagram there. Tammy, can you just unmute and talk Neil through that one?
Hi, Neil.
Hi.
Hello. I'm pulling it up here.
Fabulous. I've just had a new colour test done, and it's the first time I've seen this new result from UC Davis. What does it mean?
Right. Let me see if it attached on the email.
It was quite large actually, Tammy. I couldn't print it off. It was massive.
Right. I can send it again.
Okay.
Tammy, who is UC Davis?
It's the University of California, Davis, and they have a lab that does a lot of the testing for color genes.
Okay, thank you.
And they're also the lab that we're working with in order to perfect the Irish spotting. And so what we'll be doing is we'll be asking people who are testing their mantles or their harlequins or their merles to send in pictures of the dogs they're testing as well, so we can correlate the sequence length of the merle of the Irish spotting gene, as well as the sequence length of the merle gene. And so we can very definitively predict in the future what the results will be. I'm sorry it didn't attach to me. Neil, I did get your email.
Yeah.
But it gave me a delivery notice failure.
Okay, I'll try again.
I've sent it again in a smaller version.
Just bear with me.
Okay. Any other questions we can be chatting about while we wait for that? I try not to be very.
Well, can I ask a question, Neil? As a fawn breeder, so
yes, sir.
We've got some. Well, we've obviously got some very talented breeders here. Obviously, one is Joanne, because 2 of her mantle dogs I've given the CC to. So kudos to her. The point being is that we had a big campaign probably 20 years ago to introduce the mantle as a recognised colour. And I know it was obviously quite clear that Jean Taylor made that her ambition in order to make that happen. There's a lot of, and I don't know what the word is, negativity with regards to the Merle. And I think there's a lot of Harlequin, inverted commas, breeders that are against the Merle. Clearly the US have taken it on board. Clearly Europe have taken it on board. What's going to happen with the UK?
I think, we went about it in North America with an education mandate. And the same thing has occurred in Germany. I mean, without Frau Bachmann's, hard work, we wouldn't have the unity both at the UDDC level, the DDC level, and she's a very effective communicator, and she really has helped people understand that what we need to avoid are mating 2 animals that have Merle.
Absolutely.
And that's the bottom line. We cannot continue, and I'm sorry if I sound preachy here, but we can't continue to do Harlequin to Harlequin breeding and really feel like we're standing on the ethical ground we need to be standing on. I realize many white dogs live out very happy lives, but many of those dogs end up on the trash heap of life, and that is not fair to those dogs. So for me, the education component is the Merle itself offers us many good things, and we cannot have our Harlequins without our Merles. On the flip side, we have to clean up what we think is a morally acceptable breeding and really question ourselves if we're breeding Harlequin to Harlequin, Harlequin to Merle, or white to Harlequin. You know, those are ethically, I think, very challenged decisions. And you know, one of my motivations to do the seminar, I was delighted to be asked, was to help get that conversation started and to do it, you know, based on the facts that the science presents to us.
No, I appreciate that, and I think the big
and by the way, you know, once you fall in love with your first Merle, they're a Harlequin in a different jacket. They're absolute characters, just like Harles.
Yep. Can I ask a question, please, Neil?
Yes.
Have you read the research? Now, I had meant to look this up beforehand so that I actually knew the detail on it. But there was some research done. I think it involved something like 2,500 dogs, where one parent of each of those dogs was a Merle. And they were looking at the lifespan of it and the Merle-related problems that those actually had that had an impact on the life and health. Now, Great Danes were mentioned in that.
I haven't seen that research.
Okay, so, well, Dane's one of the breeds mentioned in that.
Huh. Do you happen to remember any of the authors' names?
No, but I've got it somewhere on email. It's just, unfortunately, I'm sat next to the whelping box at the moment because I'm rather busy.
Yep, no problem. You know, hopefully Neil Morgan will share my email with anybody who wants it. And I'm sure it might be in old Midland and West annuals anyway. I can't remember if you put emails in there or not. But please, by all means, send it to me and I'll continue the conversation. I'm quite intrigued because the only two health issues we see related to Merle are where Merle is homozygous, and we get complete blocking of the production of pigment to organs in the body that require pigmentation. You know, our inner ear functions by having these tiny little hairs called cilia, which sit in a field of pigment molecules. And as the cilia move, that moves the pigment molecules, and that's what actually the nerves interpret and how the brain analyzes sound. The same thing on the back of your eye, the retina area, that again is pigmented, and the rods and cones sit in those pigmented areas. You take the pigment out, and that area shrinks, and we have non-functioning rods and cones in our eyes, and that's why some white dogs and some Merlequin dogs are blind.
Yeah, I totally agree.
With the microphthalmia, we do see it's fairly common for it to be unilateral, and also the deafness can be unilateral, in which case you'll notice the microphthalmic dog, but the dog that lacks hearing in one ear is much harder to detect. I'm going to go check my email again now and see if that came through.
Can anybody hear me at the minute?
Yes, we can.
Yes.
I just want to know if everybody is actually going to take notice and accept what I've been trying to say for the last 18 years, that we need the Merle. So are we all in agreement now that we need the colour Merle, or we will lose our colour Harlequin?
Can I answer that just a little bit? When we go to the breed council, I would say 90% of the people on the breed council representing their clubs. I'm trying to think who are colour breeders. Most of us who attend the breed council meetings.
None of you are Harlequin breeders.
I don't know. I think there's an issue with as well that the people sat in this webinar at the moment are going to be ethical. We're going to test our dogs. We know what colours they carry. You know, you open the door and the floodgates open. We've already seen what happens when, you know, people are, you know, sort of pet people are breeding. So the minute you say, oh yeah, Merle's fine, you're going to find that the prices of the Merles go up. So Merle puppies become really expensive. And you're also going to find that maybe the ethical people use Merles where necessary or where they should do, but you're going to find other people are going to be using them and perhaps they're not going to be so careful.
Well, you don't even.
You're going to have a flood of whites.
You
Then.
don't agree with the Merle anyway, Chris. So, you know, you don't want it. So.
Has that, sorry to interject. Has that come through, Neil?
It hadn't as of yet, no.
Where's Tammy? Can I give you the email address because it's causing a
Just
problem?
wondering if
you can pop it into the chat section here if you like.
can I send it to you on Facebook Messenger?
Yes.
Right, you'll get it then.
The big question is then, because listen, there's 24 people sat around this table, which is, I don't know who's representative of the Breed Council. What are the Breed Council going to do about the colour Merle?
Exactly.
We did have a vote at the last meeting of the Northern Great Dane Club Committee, and we were asked individually what our attitude to the Merle being allowed to be shown was. Obviously, I don't know what everybody else's vote was, but I was okay with the Merle being shown. But going back to Chris's point about the fact that unethical breeders may breed with Merles, that is like saying we shouldn't have harlequins or fawns or blacks or any other colour because unethical breeders will breed from them. You cannot legislate against unethical breeders. All we can do is police our own backyard, not everybody else's. So just saying no Merles because unethical people will breed from them, you might as well say no harlequins because unethical breeders will breed from them.
But this is where we need to change the colour not recognised. We need a colour Merle first, and then when it goes to register your puppies, you've got Merle to Merle, and the Kennel Club will automatically not register them.
Yeah. If they would do that, that would be good.
We need to get the colour Merle recognised and on our pedigrees. Forget about showing for now. We need to get the colour recognised.
Right. If the KC are currently saying mating Merle to Merle is not allowed, basically won't, but yet mating two harlequins is mating two Merles together. They just happen to be modified Merles and we call them harlequin, and there is ethically no difference between those two matings.
Neil, the
and I have no problem, by the way, with the Kennel Club saying, hey, I'm not going to allow you to do this.
They don't. The Kennel Club do not say no Merle to Merle matings and will register puppies that are Merle to Merle matings. The Kennel Club will basically register any mating from any Kennel Club registered dog, whether it's
can I just come in
or not.
Can I just come in?
Oh, go
on. The Kennel Club are not saying you can't do this and you can't do that. Last year, as you know, Joanne, the Kennel Club put it back to the Breed Council and the breeders to say, what colours do you want registering? Whether you want male up there or you don't, whatever. Now, that came just before COVID. The meeting last September with the Breed Council and all the clubs was cancelled, obviously because of COVID and one thing and another. So the Kennel Club are not saying you can't or you can't do that. They've given it to us. It's in our hands to all get together and for me to put something to them and say, this is what the breed want. So we're not saying, the Kennel Club are not saying, no, you can't do this. At the moment, yes, because that's how it was. But they have given it to us to make educated decisions and go back to them.
Yeah,
And that's
well,
what we're in the process of doing.
this is why we're having Neil, you know,
Like-
explain it more now. But because
Maria.
certainly you
Yeah.
can make colour
Actually-
not recognised and
But
colour
she-
not recognised, and the
Yeah.
Kennel Club will register them.
Can anyone hear me?
Yes, I
Yeah.
can hear you, Sally.
Okay, Joe, I just want to say, you know, you're saying that if the Merle isn't recognised or cannot be shown or cannot be registered, that there will be no Harlequins. It's been Harlequins for many, many years. It
Agreed.
doesn't mean, you know, it doesn't mean that if we don't recognise it as a registered colour, that there will be no more Harlequins. Where's that
Well,
come from?
Totally
It just
agree.
gets diluted. Over time, we'll just end up with whites and more whites. We'll just end up losing it.
You know, it's just because a Harlequin is a modified Merle, it still doesn't make it not a Merle. And so arguing that a dog is fine and acceptable because it carries the dominant Harlequin gene, but suddenly it's unacceptable when it carries the non-Harlequin gene.
But that's what
I find the logic in that really difficult because, you know, from an ethical standpoint of view, we'll always have Merles coming from the Harlequin mantle family. It's a family of colors: Harlequin, mantle, and Merle.
But that's how it's always been for many, many years. You know, the Merles have been accepted as being part of a breeding program for Harlequins, but just has not been accepted as a colour. That's how it's been for many, many years. It does not mean that there will never be any more Harlequins, Joe.
That's not quite how I'm seeing it. I'm trying to say we need the colour Merle so that we're not throwing the baby out with the water, as it were.
Diversity, a bigger gene pool.
Absolutely.
Yes,
Okay.
we need the colour Merle. Oh, my back.
Okay. So can I just ask a question then? So on-
Can I hop back in and
Yes.
answer Tammy's question now?
Yes, please.
Yeah.
So Tammy, what the university lab is now doing is they show that there are 5 different peer-reviewed publications talking about the sequence length in the Merle gene and where that falls from the scale of cryptic through atypical through standard Merle into Harlequin Merle. And the particular dog you tested had a sequence length of 263 base pairs, and that sits it into the standard Merle color. If you'd had, we'll say, only 220 base pairs, or N number 220, you would have had a cryptic. And when you get out into the 270s to the 280 area, that's where these false Harlequins, these Merle Harlequins, can occur. So they're just telling you exactly what it is. You will find that generally speaking, the number is going to be a multiple of 3. And that's just because the poly T-tail, the stuttering occurs in groups of 3. So you've got 3 T's, okay, the base pair T, and you've got 3 of them. And so if in the course of it being reproduced, if it shortens up, it'll be reduced in numbers of 3. If it lengthens out, which it can do, it will be increased by factors of 3.
Okay.
So they're just giving you more detail in the information.
Fantastic. Thank you.
And it also lists on that the source papers for where the original sequence length came from. There was Clark has a paper, Langvin has a paper, and there's some papers out of the group in the Czech Republic.
Great.
Okay.
Okay. Sandy?
Well, I think it's a promise after this very good presentation. To quantify who would like us to put forward to the breed council, because it's got to come from a club. A suggestion that we can start thinking about showing the Merle.
Can I just come in? At the last meeting in 2019 that we had, we weren't talking about showing the Merle. We were talking about getting the Merle registered with the Kennel Club. Now that has been put by one of the clubs or a couple of the clubs, and that should have been discussed at our last September meeting and a decision made. We didn't get to the meeting, unfortunately, so that is still pending. The Kennel Club will do whatever we say, whatever we advise, but what we have to get right, we have to get it spot on, because once I submit this last one, that's set in stone. They're not going to change it again.
But this is just
Go on, David.
So I was just saying, but this is just, that part was purely about registrations, wasn't it? That wasn't about showing. It wasn't about changing, but it was just registrations.
Yeah, registrations.
Maria, my problem is that it's gone from the Kennel Club to the Breed Council.
Yes.
And you're saying that it's up to us breeders what we would like, but us breeders have not been asked. I only know about it from being on the Breeders and Owners, which weren't asked either about this colour Merle. And so how are you getting a open-minded view on what the Harlequin breeders want?
Because the clubs, I sent an email round and I asked the clubs to contact all their members and get the feedback from all the Harlequin breeders, what they want, and that's what we're waiting on. But unfortunately,
when is this done?
This was done last year, but with COVID, everything was put on hold, Tammy.
I personally have never had an email from any of the breed clubs that I'm a member of asking my opinion.
Because of the COVID hitting at that time, it was only, I'm talking like a day or two. Everything was just left on hold because in the meantime, more evidence may have come forward, and like this meeting and what everything, all the stuff that we just discussed, that again is going to help everybody that is against it or for it. It's going to clarify things for everybody. So it was better that it was put off, because now everyone's got a more educated view of things.
Yeah. So will I, as take me away from the Breeders and Owners Committee, will I, as a member of the Midland and West Great Dane Club, etc., will I receive an email on my opinion about the Merle colouring?
I would hope you would, yes.
Okay.
Okay.
Are we
sorry, Tammy, I don't understand why it's not been discussed at your committee meeting.
Yeah, oh, I don't know that either. Because we're not Breed Council.
We have Breed Council have asked the club to come on many times and be part of the Breed Council. For whatever their own reasons, they don't. So there's lots of things they don't get involved in, Tammy, as you know, and it is infuriating. But we can't force them.
Tammy, did you go back and ask the committee to join?
So when this goes to, is this going to go to each of the clubs, and the club then have a vote that goes to the Breed Council to carry it forward? Is that the way this is going to work?
What I asked for was, I asked for the clubs to go to their members, because they're the breeders. A lot of clubs will have a committee that no one is a Harlequin breeder.
Yeah.
And they don't understand the colours. They're just with the fawns and the brindles. So I asked.
Oh, right. Okay. Yeah. Can we bring Adam in there? Adam, go ahead.
Thanks, Neil. Maria, can you introduce yourself to Neil so he understands who you are? Because I think you've got an important role in all of this, and he probably thinks you're speaking out of turn, and I think you should understand where you sit with all of this.
Right. Yeah. Hi, Neil.
Hello.
I'm Maria, and I'm our breed health coordinator for the Great Danes. in the UK.
Right.
So
I know what that job is too.
Well, there you go. You know the problems.
So we've got 9 clubs, 8 of which I believe are active within the Breed Council. Unfortunately, Tammy's club is the one that isn't. And, the point being is here, I'm not a Harlequin breeder. I haven't heard anything about what's happening with the Merle. I've not had a letter. I'm a member of the Great Dane Club. I'm a member of the Midland and West, life member, and I've not heard anything.
The problem.
So the point, excuse Maria, just let me finish. COVID is a pain in the butt. We know that. We're in October. It's been going on since February, March. Whatever happened last October, November, we've all learned to adapt. And this very session here is testament to that, is that we've got a Zoom meeting. So if the Breed Council haven't adapted, something's wrong. If the Kennel Club haven't adapted, something's wrong. So what's going wrong?
Well, the Kennel Club, I don't know if anyone's been trying to go on there and get some information from anything. That's a big no-no. The system's all being updated and you can't find anything. You can't even find your own dogs that you bred.
Yeah,
so
from
the Kennel point of view, that's you're not going to get anything from there. This all happened, and I'm saying it, I've just said it was last September. It wasn't, it was early on this year. So just before COVID in March, when we should have been having a meeting, that's when evidence come, and the Kennel Club said, what do you want to do? What do you want, what dogs do you want registered, i.e. the male? We haven't got a problem with any of the other colours. It's only when it comes to the Harlequin, which is a minefield on its own. So everybody needs to have a look at this, all this information, and the breed, well, a lot of the breeders know anyway, and then make a decision. The clubs can't go to the Breed Council meeting and give a decision and say, we don't want male registered, or we do want male registered, or whatever. They have to go by the breeders and the members of the clubs, in my opinion. So and that's where we're at. We're waiting for opinions from the breeders.
But if the clubs aren't advising the members of the direction of the club, then what are we expected to understand? You guys, we're all members of your club, so you guys have got to inform us of what's being discussed, what's the general consensus of opinion, and then let us make a decision based on our individual understanding.
So everyone makes an educated decision. We didn't get that far because this just started off in this year, which was great. It was exciting. Where are we going to go? And then obviously, I know everyone blames COVID, but it genuinely did. It was just before COVID hit that we were into this. So we will pick up where we left off. I mean, now we can do Zoom. Maybe the Breed Council will get together and we'll have a Zoom meeting. But before that,
Maria,
we'll have to go out to the clubs.
Maria, maybe or will, because maybe is COVID's been going on since March, I'm afraid, and we're in October and it's getting worse and we all feel it. Listen, we're all in this together. But come on, we've got 9 Great Dane clubs. I'm on a general committee here of an all breed show. We've adapted, we've changed, we understand the different ways of working. Zoom is not a new experience for me with it is for the Midland and West, but this is the first one, and it's October. I've been on Zoom.
Well, Zoom's the first. This is the first Zoom I've ever done. If I don't know you could have seen me, I wouldn't know me.
Because half your face is missing.
I was just going to say, you can only see your eyes, miss, and up your nose.
But, listen.
I've got the puppies.
Come on, I know, Maria, and it's great. Listen, you'll make a mint out of all those puppies. Back in April.
Charging $3,500. What do you reckon?
Don't undersell yourself.
No, I won't.
Sorry, Janet, did you want to ask a question, please?
I forgot what it was now. I was just following up on the fact that a lot of people out there aren't Harlequin breeders, and perhaps they don't care so much about the fact that what's going on amongst that colour. I mean, for instance,
they didn't understand.
For instance, there's 24 people here on this chat today. Where are all the others? I'm really limited.
Maria,
hang on, I haven't finished yet. Thank you. Also, we're worried about the registration side of it, like Neil O'Sullivan pointed out. But by the time they get to that stage, those puppies are already on the ground. They're already alive and breathing.
Neil, you wanted to come in there. Neil?
I thought it might be helpful to tell you the way we approached it from the Great Dane Club of America perspective. I was on the board, and I said, I think we should poll our membership. And my suggestion would be that the Breed Council send out to each club a request that they poll their members about it. And, by all means, include the PDF from today to help people educate themselves about their decision making too. But I would certainly say, you've got to get to the membership of your clubs and ask them their opinion and trust them. Your members will actually work very hard to make very informed decisions.
That's exactly what I've done in March, Neil. I wrote to the clubs. We've got to go to the members, get the feedback, educate them, and then come to an educated decision.
Can I just
I've not heard anything about that.
Can I just put a note of caution before asking the majority of members? They need to be educated, because the opinions of certainly fawn breeders or non-breeders at all are that Merles are a dangerous thing, and they've been told for centuries not to breed with Merles. And we need to be able to they should all have Neil come along and tell them all about the fact that a Merle is a Harlequin, a Harlequin's a Merle. Because most breeders, non-Harlequin breeders, do not get it. So you're going to get a false result from a poll that you did without any education to a big majority of breeders. Most breeders, especially very sort of new breeders, are just going to go back and go, oh, no, you can't breed with a Merle. I've been told you can't breed with a Merle. You need to get it across to them exactly what we've been sat listening to tonight before a poll. Otherwise,
what I'll suggest is that we have a seminar just on this, and everyone can come to the meeting, get educated, and then air their views.
Yeah, I think that's the safest way to do it. I don't think you will get a true result, an educated result, just by giving a poll out to the clubs to give to all their members. I think it will show a very uneducated response from 90% of fawn breeders and non-breeders.
Yeah, well, it's something we've got to do.
Sorry, and can I just clarify, what are we actually voting on here? Are we going to be voting on, or at least giving an opinion on whether we are registering?
Yes. Yes.
We are not. This at this point, this has got nothing to do with showing.
No,
nothing to do with showing at all.
We've got to be able to register them first before we can show them. So that's got to be the starting point, hasn't it?
This has got to be a recognised.
That's where you want to go. You want them to be shown.
No, I'm not saying that. What I'm saying is we need to know whether they should be registered. Showing is something different. Before you can show, we've got to have them registered, and you've got to be breeding from them and know what you're doing. Then any showing would come later. But if you're going to register them, then I'm sure showing will follow, because you're using them in the breeding program anyway.
Yeah.
So you need to know what you're using and get that out there as you do the Harlequin.
Maria, doesn't that go hand in hand? If they're become registered, they will then be able to be shown. The Kennel Club register them and then say, oh no, but you can't show them.
No, because the registration
along with the education that people
got it in the standard.
Think that needs to be made clear, because it's all about the standard. We've already got, if my memory serves me, that it says that a blue dog can have a black nose in our breed standard, and I'd love to know how we manage that.
Yeah.
Not genetically possible.
And going back to breed clubs not letting people know. You know, let's look forward, not whinge about what hasn't happened. Let's, you know, this Zoom meeting has been great, and I agree with Sheena that people need to be educated before they can make a decision. So let's move forward, do something.
Yeah. Yeah.
So
What we've done tonight.
I know Wales were going to poll everybody.
That the clubs are not asking our opinion as members of the clubs, and this decision will get made. And as Maria has already said, this is it. Once we make another change, they're not making any more changes. So we as members of our breed clubs need to have involved in this decision, and we haven't been.
Totally agree.
Wales was planning to. We just didn't get a chance to because of COVID. I say that hand on heart, being a Harley breeder.
Stop using COVID as an excuse.
Move on. It's all right to win. It hasn't happened. Let's move on. This is, you know, let's do something. But people do need to be educated. And I think having a meeting where people can come and be educated, like we have today, would be absolutely brilliant. I just hope to God that they turn up, unlike our health day. You know, this is what worries me. We put on all these health days and hardly anybody, big breeders that should know better, don't even bother to come. They don't even represent our breed. So I hope they do for the colour, because they certainly haven't done health.
Just a question
Well,
to Neil. Neil, can I just ask you a question? How you proceeded in America? Did you go for registration first and then showing afterwards, or did you go for both at the same time?
You could register a Merle as a Merle in the US. So what we had to do was, you know, write the change into the breed standard, get membership approval, send it to the Kennel Club, get Kennel Club approval, and then if there were any modifications, which there were, it came back to the membership, and the membership again approved the revised wording for Merle.
Okay. I believe
we have perhaps the opposite problem to you. You can register a Great Dane in America that has 10 points, that is piebald, that is chocolate. You know, all colors that are due to a gene, foreign to our gene pool, but yet, unfortunately, they'll allow them to be registered. And you really find the unethical people pounce on that and purposely breed chocolate harlequins and blacks and blues with 10 points.
Yeah, well, we have them as colour not recognised over here. And
I'd prefer that terminology.
Can I ask another question? If we're saying, or, you know, a lot of people here want to go ahead with the registering of the Merle, I have to say I'm still undecided in some respects. Are you going to have Merle, or are you going to have Mantle Merle, or both? Are we going to have two Merle colours, or is the standard going to say that it can be Mantle or it has to be solid colour?
So if I can address that from a genetic point of view,
Yeah, they're different.
Because black and because mantle are accepted colors in the breed, when the Merle gene is imposed on either, you're then going to have a black will become a solid Merle, and a mantle will become a mantle Merle. So you really have to recognize both. And that was a big discussion in the US too. But I was able to show, again, you know, through education at our annual meeting, I was able to show that you can't suddenly discriminate against, we'll say, the solid Merle and say that the mantle Merle is the only one accepted. Because there's nothing wrong with mating a black to a merle, even a mantle merle, but then you'll get solid merles, and those solid merles are just as acceptable as ones that have the white pattern from the mantle.
Don't confuse them anymore, for God's sake.
Go for it, Adam.
Sorry, it's always me that asks questions.
Yes, Adam. Yeah.
Well, so clearly, obviously, we have our colours defined by colour in the UK.
Yep.
In Scandinavia, they have black or white, which allows piebald. So, Neil, as a non-Harlequin breeder, what is the advantages, disadvantages of that colour piebald?
Right. So.
Well, hold on a minute, Chris. Let's just have an opinion here, because I
was asked to attend a UDC meeting a couple of years ago where that very point, I was basically asked to come and discuss that for the UDC. It is speculated, that piebalds first started to occur in German lines post World War II. People in the UK were not seeing them prior to the Second World War. And if you ever had a discussion with Bill Siggers, he would tell you the first piebald dog he ever saw, who was treated as a Harlequin, but he was piebald. And the speculation was the color was brought in from another breed, just like black and tan is foreign to our breed. It occurs, but it's foreign. Same thing with chocolate. So I thought it was a very bad compromise that the Europeans made when they the FCI standard through Germany was changed, basically allowing it. And I speak enough German to understand how the phraseology can be passed as acceptable. So like a mantle is Plattenhund, right? Whereas Glefleck is very definitely the torn patches, and the piebald term is the smoothen patches. That was a very
fluent, Neil.
Huh?
You're almost fluent.
Oh no, not if we're talking finance.
So how can these people be showing a piebald in Scandinavia?
Oh, you can show it in any FCI country right now, but the question is it correct? And I argue it's not. I argue it's a color that is foreign to our breed. It is no different from saying, I get chocolates start cropping up because I have American fawn and brindle breed, and now I want chocolate approved. It's not correct.
Can I just say one point here, please, to 24 people? We've got somebody, thank you, Neil, that knows what they're talking about, who can educate us all, that obviously needs to sit in front of the Breed Council and educate the Breed Council. Whether they agree with it or whether they don't, that's their interpretation. But at the minute, we've got a very, very fragmented opinion of Merle, opinion of piebald, and I threw that one in just as a throwaway. A very, very fragmented opinion of Merle, just like it was a mantle 25 years ago. And unless we've got somebody sitting in front of the Breed Council who is our representative to the Kennel Club, then we'll never move forward. This lot here, this crowd here, can talk about it till the cows come home. It isn't going to happen.
Okay, everybody. Okay, can you hear me now?
I agree with you, Adam.
Thanks, Sharon.
Yeah, can you hear me?
Yes.
We can hear you, yeah.
We've got to wind this up now. So thank you for everybody who's taken part and listened, and thank you so much, Neil, for giving up your time for us all and educating
My
us.
pleasure.
I think
Thank
our
you.
way forward is for each one of our clubs that are part of the Breed Council is to have a look at the colour standard that the American Kennel Club are using.
You have to, Sandy.
Put something together to poll our members, and then put that forward all the clubs in the same sort of way.
Sandy, can I just ask a question? If we use the American Kennel Club standard on Harlequin, it's the only standard, I believe, in the world that actually asks for the white neck and front, which means that in order to get that, we're going to have to use mantle in there to come in from the SLOCI.
Chris, I'm not talking, I'm just saying to look at it to get some idea of what we're looking to poll our members for. And then we'll see from our members, and I think when we poll our members, we need to find out if they're colour breeders or fawn and brindle or whatever, or so we have to find out what the majority of Harlequin breeders want. And then
can I just say something, Sandy?
Yep.
I think Sheena made a really valid point that the lack of education with a lot of non-Harlequin breeders, those of us that have attended today have an interest in it. It was open to a much wider audience and they didn't choose to take it up. So how are we going to educate them? And if we don't just want to put a poll straight out there just to the world and his wife, and
we'll put something together. The clubs should put something together and liaise between us and put something together which sends out the PDF together with the poll, so that whoever we're asking to contribute, if they want to contribute to the poll with their opinion, they'll be able to see the PDF which will explain it to them. Other than that, I will also think that Maria will probably like to organise, when we do have a Zoom meeting, as hopeful with the Breed Council, that we can invite Neil to repeat his presentation.
Yeah.
I'd be happy to do that.
Yeah.
And I'd be happy if any other breed club wants me to go through it for them as well.
That would be great. That would be great. It would also be nice to sort of leave this on a positive note, because the Breed Council did get the mantle accepted 4 years ago, and hopefully, all we can do is see how everybody feels about this, and if we can get a seminar or something where everybody can come, and Neil would be kind enough to,
Liz just pointed out, Liz Cook, our treasurer, that we will be putting this PDF, Neil's presentation, and this recording onto our Midland West website. So it'll be there.
That's great news.
Okay, so we're closing the meeting now. Okay, thank you, everybody.
Thanks, Neil.
Thank you.
Both Neils. Both Neils.
Okay.
Shut up.
Okay, bye, everybody.
Bye.
Thank you.
Bye.
Bye.
Bye, everyone.
Bye.
Bye.
Right, what are you doing now?
What do you do now? Am I off? Am I off, Tammy? Am I off? Can you hear me, Tammy?
Okay, can I see those puppies one more time, please?
Hang on.
That's cheating.
I know, I just want to get my puppies back. My little puppies.
That poor woman looks like she could fall asleep while she's